Welcome back, everyone. This is the third in a series of lectures in Chapter 5, and we are now in Part 2 of Section 5.4, which covers the endomembrane system, focusing on a phenomenon called protein sorting and targeting in the different organelles, but particularly in the ER. We only have a single objective, learning objective for this video, and that is to predict from the presence of various signal sequences whether a protein will be translated on free ribosomes or the rough endoplasmic reticulum and its ultimate cellular destination. So I hope that by now you have come to appreciate the importance of the endomembrane system, not just in the ability to manufacture biomolecules like proteins, lipids, and carbohydrates, but also in their exquisite ability to process um, these biomolecules in order to deliver them into the right places in the cell in order for them to perform their very specific and important functions. You can, of course, infer that any kind of aberration or problem in terms of the delivery process would actually cause substantial harm to the cell. In fact, there are a huge spectrum of human diseases that are associated with simply this defect in the ability of certain cells to deliver proteins or lipids and other materials through the endomembrane system to their final destination. One example of this would be Parkinson's disease, and there's a whole array of cancer um, states that are simply due to failure to deliver proteins, for example, in their respective destination. So this phenomenon is called protein sorting that we will be focusing here. Um, of course, there are also other ways by which other molecules like lipids also regulate their transport, but we will be focusing mainly on protein sorting here. And protein sorting is the process by which proteins end up where they need to be to perform their function, according to specific amino acid sequences called signal sequences. So there has been a general pattern that was observed throughout the years, and that's for proteins with no signal or no signal sequences, they actually remain in the cytosol as shown in A. And if they have an amino-terminal signal, then they get targeted to the chloroplast, to mitochondrion, or to the rough endoplasmic reticulum. So you can see that the presence of an amino-terminal signal, which is very specific for different kinds of proteins. There are several different kinds of amino-terminal signal. So some amino-terminal signals, they are actually recognized by specific molecular machines that we don't have to know at this point. To deliver them to the chloroplast, some amino-terminal signal sequences would be recognized by a different molecular machine and deliver it to mitochondrion. And then, of course, the ribosome itself sometimes would deliver it if it has another amino-terminal signal sequence to the rough endoplasmic reticulum, as we will see in the next few slides. Now, there are also signal sequences that are found within the polypeptide of the protein that is being synthesized by the ribosomes. And these particular polypeptides are targeted to the nucleus. Specifically, it is important to mention here that this internal sequence contain a so-called nuclear localization signal, or let me see, NLS, nuclear localization signal, which is recognized again by a molecular machine that allows the protein to move through the pores, the nuclear pores of the nucleus. So the signal sequences, therefore, are intrinsic in the amino acid sequence of the polypeptide itself. So it's not something that is added to the protein, but it is part of its native sequence. So let's have a closer look at one of these protein sorting pathway or protein transport pathway, and that is protein transport or sorting via the rough endoplasmic reticulum. So if a protein that is being translated by the ribosome has a specific amino-terminal signal sequence, then it gets targeted to the ribosome, but it ends up in three possible locations. Okay, one is within the lumen of the endomembrane system right there, or it could be embedded in its membrane and become what we call as an integral membrane protein or a transmembrane protein, or secreted out in the cell, okay, uh, from the lumen all the way. So if the protein is destined for the lumen or will be secreted outside the cell, the protein is actually fed into the ER lumen as it is synthesized by the membrane-bound ribosome all the way. Now, if the protein is destined to be embedded in the membrane, it is inserted into the membrane as it is being synthesized. So we call this as a co-translational insertion process by the ribosome. So let's have a closer look at how amino acids that are destined for the lumen are actually delivered there in the endoplasmic reticulum. So the main critical signal here is the amino-terminal signal sequence in the growing polypeptide chain during the translation process. So just to introduce you to how translation is happening, we haven't really touched yet the molecular mechanism of translation, but it suffices to say at this point that when a polypeptide has just started getting translated, it is the amino terminal end of the polypeptide that first comes out of the ribosome. And so when this amino terminal end has a signal sequence, it is immediately recognized by a special protein complex called SRP. It's called the signal recognition particle. It binds to this signal sequence in the amino terminal end of the growing polypeptides, and it stops translation for a moment, for a brief moment. And during that pause, this SRP will then in step B bind to what we call an SRP receptor. Your figure in your textbook mislabeled it. It's here it's showing as a phospholipid, but actually this is the SRP receptor. So the SRP bound to the amino terminal signal sequence delivers the elongating polypeptide to the channel in the rough ER by its binding to an SRP receptor. So two binding events. So the SRP first binds the amino terminal signal sequence, followed by its binding to the SRP receptor, and therefore positioning this elongating polypeptide or growing polypeptide right in the opening of the channel in the rough endoplasmic reticulum. Then in step C, once the SRP receptor brings the ribosome to a transmembrane channel, delivering the elongating polypeptide chain, the SRP will now dissociate. It has done its service, and protein synthesis resumes. and the growing chain, polypeptide chain is threaded through the channel like so. And the N-terminal signal sequence is cleaved by a special enzyme called a protease that is located inside the lumen. And so once the ribosome has terminated translation process, they all gets disassembled. But now the freshly synthesized polypeptide is now delivered completely inside the lumen. Now, this protein inside the lumen may either be transported to the lumen of another organelle, let's say lysosome or other organelles, or it may be secreted out of the cell, or it may even remain in the lumen of the endoplasmic reticulum itself. It all depends on whether it receives additional processing or modification, which determines its ultimate destination. Now, the second case for transferring or targeting polypeptides from translating ribosomes to the rough endoplasmic reticulum is when they have a different sort of signal. So instead of an amino-terminal signal, the polypeptide chain being translated by the ribosome actually carries what we call as an internal signal anchor sequence. When this happens, the protein has a very specific destiny or destination, and that is that it becomes what we call as a transmembrane protein. So this is the figure here depicts how transmembrane proteins or integral membrane proteins gets targeted during its synthesis in the rough endoplasmic reticulum. So first, what happens is that the ribosome in the cytosol starts translating the polypeptide, and as it translates it, eventually there is an internal amino acid sequence that is called signal anchor sequence, which then allows this sequence to bind or interact with the channel in the rough ER, like so, right here. And when this happens, the signal anchor sequence causes the channel in the rough ER to open up. And because the signal anchor sequence, it turns out, is very hydrophobic, here we go again, hydrophobicity. Because it is very hydrophobic, it can easily slide and dissociate away from the channels in order to penetrate the hydrophobic region of the phospholipid bilayer. So the ER channel therefore releases the protein into the membrane while it's still being attached to the ribosome and being translated. So the ribosome will continue to translate this polypeptide chain into its complete form and then dissociates itself. But once the translation machinery disassembles, the protein now remains integrated into the membrane. So it's a pretty cool, pretty nifty stuff. Eventually, these transmembrane proteins would serve as channels, pumps, receptors, or enzymes. So this concludes section 5.4. And before you leave, please make sure that you answer this RIF question. So if a mutation blocked the function of the signal recognition particle, or SRP, making it unable to bind signal sequence, what would result? A, all proteins would end up in the mitochondria or chloroplast. B, all proteins would end up in the nucleus. C, all proteins would be secreted from the cell. Or D, all proteins would be translated on free ribosomes in the cytoplasm. So figure that one out. Good luck. So I'll see you in the next section, section 5.5, which should be the last lecture video for chapter 5. Thank you.